Welcome to Genetics as the Soul of Precision Medicine, a conversation with Dr. MaryClaire King. I'm Janet Rossant, and I'm President and Scientific Director of the Gairdner Foundation. Dr. King is the recipient of the 2021 Canada Gairdner International Award for transforming cancer genetics and oncology with the discovery of the inherited susceptibility to breast cancer due to the mutation of the BRCA1 gene. Dr. King's research has had an impact on millions of women and families around the world, and I'm so happy to have her here today to tell us her story. Today's conversation is part of a series of events celebrating Gairdner Science Week. To see the full schedule of what's coming up, visit the schedule section in Hopin or visit our website at gairdner.org. So thank you for joining us, MaryClaire.
Hello, Janet. It's so good to see you.
It's great
Thank you for inviting me.
It's great to see you too, and to see you wearing your Gairdner pin, which I am also wearing mine.
So yes,
it's very nice.
I was never a member of a sorority, but now finally I have a pin.
You are a very eminent group who share that pin. So, and I say you received the Gairdner Award this year for your work on genetic predisposition to breast cancer with identification of BRCA1. Tell us a bit of the story of that discovery.
Oh, sure. Sure. I got interested in cancer after my PhD dissertation, which indeed was in human evolution, a completely different field. And when I was offered what we would now call a postdoc by Nick Petrarcas at UC San Francisco, he was interested in a problem that went all the way back to the ancient Greeks. And of course, he was Greek, so he had actually read this properly. And that was the observation that there are families in which breast cancer appeared to be far more common than one would expect by chance. Of course, nowadays it's a very common cancer, but even overlaid on that common cancer, there were families that were so severely affected. And these families, apart from their breast cancer, were the healthiest, most productive families imaginable. They did not have any extraordinary environmental exposure. It was a mystery why this was true. And this had been noted by Herodotus. This had been noted by Paul Broca, working in 19th century France. And then very systematically, it was organized. The data was organized by Jane Lane Clapon in the British Home Office in the 1920s. And what Dr. Lane Clapon showed was that women who died of breast cancer were far more likely than women who died at the same ages of other causes to have mothers or sisters who had died of breast cancer. Mm-hmm. Now, she showed the same thing for men and lung cancer. And her hypothesis for the latter was that these men had shared environmental exposures occupationally and had shared smoking. But as she pointed out, breast cancer was much more of a mystery. So why was this happening? And very good epidemiology from that time until the 60s reinforced the validity of those findings, showed that the finding was worldwide, that there was a familial clustering of breast cancer, reinforced that it was increasingly severe amongst women who developed breast cancer early and in families where there was both breast and ovarian cancer. So Dr. Petrarcas said to me this seemed to him to be a problem. He gave me carte blanche to try to sort it. And I thought, if we are truly desperate, if we can't think of any other explanation, this is like, the purloined letter. Mm-hmm. If we can't think of anything else, maybe it's genetic. And if it's genetic, we need to do two things. We need to sort out the way in which it's genetic. That is, we need actually some parameters of how the hypothetical gene or genes would be behaving. And then we need to actually find the genes. Genetics is not fantasy. There is actually a physical reality behind every statement. And if I could do that, I would have proven the case. To be honest, it never crossed my mind that it would have the impact that it has. I was an evolutionary biologist.
Yeah. So you mapped it. I mean, it sounds easy today because,
I know. My undergraduates say, well, that's nice. Then what?
Genomes left, right, and center, but it wasn't easy. I mean, this was really hard work to get even to the sort of location on the chromosome.
There was no map.
No, no.
We built the map as we went along.
Yes, as you went. Yeah. So it's really, but it was a fundamental finding, and of course it has had ongoing impact. Today it continues. BRCA1 and then BRCA2 are major drivers of breast and ovarian cancer. Did you ever think it was going to have such an impact?
I was not the one who realized the impact. I just wasn't. I mean, I'm not a physician. I in those days did not think in those terms. I've learned to think in those terms. But I came out of evolutionary biology. I came out of math, for goodness sakes. Right? And it was Bernie Fisher, the extraordinary surgeon who really was the pioneer in reduced surgery for women with breast cancer, who said to me after we mapped the gene in 1990, he said, you're not going to be able to control what happens with this. He said, once this gene is cloned, this is going to be far bigger than you. And I said, yeah, sure, Bernie. Yeah. I said, it's a few families. He said, this is not a few families at MC. This is really going to have major, I think he said, this is going to have ripple effects beyond what you can see. And he was absolutely right. And it took that perspective of a physician actually seeing patients to realize. And no, it was extraordinary. It was extraordinary.
Well, still, you started down that path and you set everything in place for everything that followed. So it's really amazing. So the Gairdner Award, how important was that? Was that a great?
It was just totally fabulous. It was just great. So I have to share a secret. So I am incredibly proud of receiving this award, and particularly so because, oh gosh, probably 15 years ago, maybe longer, Gairdner Foundation gave this award to the guys, and I use the term advisedly, guys, who were responsible for the Human Genome Project. And these are my brothers in science. I mean, these we all play together all the time, right? And so I think there were 7 or 8 of these fellas. And they all came to Canada, and they spread out all over your fabulous country. And they were from St. John's on the one hand to Vancouver on the other. And they gave talks, and they had a fine time. And of course, several of these people are in my department, Maynard Olson and Phil Green and Bob Waterston. And they and all the guys came home, and then they all met up here for reasons that I can't recreate, but they met up all here in Seattle, all 8 of them. And they just went on and on about the fine time they'd had with the Gairdner. And I thought, oh, geez, oh, boo, oh, golly, wouldn't it be nice? So I am so honored, but I'm so bummed that I can't come to Canada.
I know.
I hope you'll let us come some year when it all settles out.
Well, that's certainly the case, and you're not the only laureate. Last year, of course, was the same. We also went virtual, and this year, and I must say, every laureate is saying the same thing. What we know about the Gairdner, and what is special about Gairdner is the fact that we don't just give awards. We really bring you to Canada. We like to engage you with the faculty, with students, with high school students. We'll still do some of that. We'll do it virtually in these kind of virtual conversations.
And of course, the high school students, they'll have it all over us because they're completely facile with this stuff.
Yes, well, they will ask the very tough questions. I sat in on a high school lecture by, guess who? David Julius. Yesterday, the day after the Nobel Prize, he did a lecture for us. But the high school students and the younger students too, they ask the very best questions. There's no question about it. So.
You have in Canada very good elementary education in science, and it shows. I mean, pre-COVID, of course, I was in and out of Vancouver all the time, and there's just no question that your education in science for your kids is just terrific. I hope you'll arrange for a general invasion of Canada by all the Gairdner Award winners going back all the years of COVID, and we'll all come.
Yeah. Yes. We hope to do that. We'll have to gather some money to.
Yes, exactly.
But we certainly want to do that. And I recognize how everybody is missing. We're missing you too. Right. It's not the same without meeting people.
It's not the same. It's not the same.
Yeah. Anyway.
And you and I could even meet. Such a concept.
Yes.
I could visit your lab. I mean,
We've met virtually several times in various formats, but I don't think we've actually met in person.
It's nuts.
We should do that.
Right.
So let's get back to science. I've got some sort of science questions here. There's one I always ask, and you've named some of the people you worked with and so on in the early years, but if you think back, who were the role models? Who were the people who really put you on the path to science and success?
Right. Well, I think of this question as having two layers. The first is the literal answer to your question. And those would be my teachers who really helped me realize that there was such a thing as a scientific career, because I did not grow up in a scientific family at all. A very loving, very intelligent family, but no higher education, certainly no concept of what a PhD in science was. And I would say Alan Wilson, of course, my PhD advisor, Kurt Stern, the person whose genetics course I took and never looked back, and then Luca Cavalli-Sforza, who helped me make the transition from population genetics into molecular genetics, a transition he had made. And those three gentlemen were just wonderful to me. They were accepting and open and respected that I cared enormously about genetics, and were incredibly patient with the fact that my hands were terrible because I had not done experimental biology at all. But in terms of the people that inspired me from the time I actually realized, yes, I want to try to do this as a field, I think it's the women of our generation and the generation just older. And I have to tell you, you are absolutely one of them. I mean, the elegance of your work, I mean, from the first days I read of it, I thought, oh my God, I will never be able to do this, because it takes a level of understanding of cell biology that I simply do not have. But its elegance and it's just transparent, and one can read it and understand it immediately. And I was just in awe, and I thought I need to learn to organize experiments like that and to write like that, so I can be that clear. And other people that I thought of in the same way, of course, Janet Rowley at the level of chromosomal biology, Maxine Singer at the level of how to actually carry out very good science, but also not be afraid. One of my favorite photographs in all of the modern history of science is Maxine Singer sitting with the guys in the first days of recombinant DNA down in Asilomar, and all the gentlemen leaders of our field are hanging on her every word. And here's this young woman who is confident and clear, and she's just terrific, and she's still terrific. So I think those you three, Nancy Hopkins, Joan Stites, I mean, the fact that there were women of my generation, or in the case of some of the people, obviously not you, a little older, who made this a path that we could see, that made sense for us to follow. Yeah. And they're still with us. That's the joy of this. They're still with us.
Yeah. So yeah, I think for all of us, role models, mentors, they can be male and female. The female ones do play a special role. It is that sort of being there, going ahead, being there for you, so that being in fields where they lead the way, it's much easier for other women to follow.
It also gives one a sense of how to fit in the children.
Yeah.
I mean, how each of amongst the women who have children, how they did it and how they fit it in, and that it's okay to spend 2/3 of your salary on childcare, and all those things. It's very practical. Back when I was first elected to our National Academy, so this was, I think, about 2005. At that point, all the women members fit in one room, and so we all fit ourselves in one room. And I can't remember what the stated order of the day was, but we ended up talking about childcare within 2 minutes. It was just, how do you manage it? And of course, all of us by then, our children were grown, but how did we manage? We do have challenges, and it's helpful. It's enormously helpful to see how others have managed it.
I think that's a more general challenge still for women, faculty, and all the rest of it. Is one of the simplest things that can help is having childcare on campus. And
it's huge.
It's just, there's still so many places that don't.
We finally do here, but it's not nearly big enough. I mean, you have to know years before you're going to become pregnant to sign up on the waiting list. It needs to expand. And of course, I mean, it's never been more clear than during COVID. I have young physician scientists in my group who, of course, their practices continue. Their patients continue to come in, and they have small children. And fortunately, they have heroic husbands. But I do not see how they are doing it. I just can't imagine with childcare closed. I mean, it's just now opening again. I don't know how they did it. And grandparents were terrific. Husbands were terrific. It's still very tough.
Yeah, I mean, the COVID has been obviously a big pressure on many aspects of many people's lives, but women across the board have again suffered considerably, and it's mostly around the children. It's very difficult. Hopefully we're coming out the other side. Yeah. Anyway, back to science. Well, actually not back to science. I want to sort of move off because MaryClaire King is more than a geneticist, more than a scientist. I sent you questions ahead of time. I called you an activist. Is that a fair thing to say?
It's fair.
Okay. So
I wear it proudly.
Yeah, it seemed to me that looking at everything you've done throughout your life, you've always been there. You're always going to be at the forefront. You're going to fight for what's right from Vietnam War on. Your human rights work in Argentina is very well known. Do you want to tell us a little bit about that story too?
Sure.
Fascinating.
Right. This was work with the grandmothers of the Plaza de Mayo. The question was how to identify their children who had been kidnapped and the parents of those children, so the adult children of these grandmothers murdered during the dirty war in Argentina between 1975 and 1983, and then how to restore those children to their families. I was asked to participate in this by our American Association for Advancement of Science, whom the grandmothers had approached in 1983, I think, maybe a little bit before. I first learned of it in 83, with this question. I had lived in Chile during the Unidad Popular, the Allende government, in the early 1970s, so I was accustomed to the Southern Cone. I can teach in Spanish, slightly fractured Spanish, but certainly can hold my own. And so when the grandmothers went with the question I just described, how do we identify a child whose parents are dead, some of whose grandparents are still living, and who is being held somewhere by militaries or collaborationists of the militaries? How can we identify those children? If we, the grandmothers, have a suspicion who a child is, how do we prove it? When they went to AAAS with that question, and AAAS turned to me, I said, I don't know how to do it, but I think I can figure out how to do it. This was in 83, and then my first trip to Argentina for the project was, or what became the project, was in June of 84. And this was, of course, way before DNA testing or any sort of DNA analysis was being used for paternity or indeed much of anything else. Paternity testing was being done with blood groups and with HLA, and what I introduced was the idea of grandpaternity testing. Simple Bayesian statistics, you can extend paternity testing to grandpaternity testing, and of course the realization that in the absence of the parents, with the parents deceased, and with hypothetical grandparents, one would need much more precision in genetic markers than one would need for straightforward testing of parents themselves. So that meant that we needed to type HLA markers, which we were able to do because there was a very good HLA testing lab that was being used for tissue transplantation in Buenos Aires, and so we asked them to help, and they were excellent. And then we were successful. We were successful right off the bat, and because we were successful, the project began to blossom quite quickly, and it was very quickly the case that we were asked to try to address the question of proof of identity for children who had been found in the hands of militaries, but for whom not all four grandparents were still living. Mm. So that led me to apply mitochondrial DNA sequencing. And this was by hand. This was one base pair at a time. Supermarket coding barcodes with one base pair at a time. But mitochondrial DNA is so informative for maternal lineages. Mm-hmm. And that was already clear this was early 1985. It was so clear. And Alan Wilson, who of course had been my mentor for my PhD work, and I was, just across the street from him at Berkeley, was enormously helpful in helping me set up. So we were able to do this for the grandmothers. I was able to they had sent me home with after the first success, the identification of Paula Logares in Christmas of '84, and they'd sent me back to Berkeley and said, "Look, Nina, you've done the easy cases. Now we want you to do cases where we don't have all the grandparents still living." And typically in those cases, we had the mother's mother, or perhaps sisters of the mother, but a maternal relative, a purely maternal relative, regardless of whether the kidnapped child was a girl or a boy. And so I was able to call them in a couple of weeks and say, yes, I do have a way to do this. And that really opened the floodgates. And we were able to identify, now the grandmothers have identified, I think it's 136, 137 children, who of course are now adults. Mm-hmm. The children grew up and came looking for them. The National Genetic Data Bank is flourishing in Argentina and completely autonomous. They honor me with asking me questions from time to time, but scientifically they are completely autonomous. This last Christmas, a member of the data bank came here to Seattle in the midst of COVID. She got special permissions to do it and took back to the data bank in Buenos Aires samples from 800 people whom we had sampled and extracted DNA over the decades, but those people had died of natural causes, elderly people, and those are now in the data bank and are being fully sequenced. Mm-hmm. And of course, the United Nations Forensic Anthropology team was formed by young Argentinians and is now the International Forensic Anthropology team for identifying victims of human rights abuses worldwide. So the main thing this taught me is that the most important questions are asked by people on the front lines. Mm-hmm. Mm-hmm. Right? And second most important thing is that if, as a geneticist, I think I can figure out an answer to the question, I should just say yes.
Yeah, well, I think that's to me what I find inspirational is that you saw the problem, you knew that it was a huge issue for these families, and obviously the ongoing issues of forensic anthropology are just enormous in these conflict zones. Right. And if you can do something about it, you should do something about it. So I think that's fantastic. It is inspirational. So how do you try to get people to follow in your footsteps? People come into your lab. Do they get sidetracked on forensic issues? How does this all play out in your sort of intellectual environment?
Well, I think it's like your lab. It's like any good lab. People come into the lab and they have ideas for projects they want to do. The forensic work, I must say, has been the dissertation project of only one person. And she is indeed a forensic scientist. That's because there are so many elements of that project over which we have no control. And so it doesn't make a good dissertation project, except in the capacity of a person who is then going to go back and work for and be a forensic scientist. People come into the lab. Most of the people in my lab are doing projects that are of medical interest, either in breast cancer or one of the other areas we work in. And then something will come up, and we need people with particular skills, and they just say yes. Your capacity to think expands to fill the number of questions that you're asked. We don't have a finite capacity for thought.
That's fantastic. So a long career from math to evolutionary biology to cancer genetics to human rights work. It's simply amazing, and you're still so full of many questions still to answer, right?
Absolutely.
So I have to ask you, how would you advise young scientists going into just anything and into science today? What's your best advice?
Right. I think there are a couple of things. One is to do as much math as you can for as long as you can. I originally thought I would do math, that I would try to do math professionally. And I majored in math in college, in fact. But I realized by the time I was in high school, I realized that there was absolutely no way that I was going to be able to be a professional mathematician. And I realized it because my brother, who is younger, was so much better at math, and he did become a professional mathematician, that there's a distinction between people who love math and like solving problems, me, right, and people like Paul who really have incredible talent in this area. Mm-hmm. But that said, I mean, I was a B+ math major. I was not a great math major. But I learned how to solve problems, and I learned how to tackle mathematics. And if you can tackle mathematics, you can tackle anything, because it is the hardest thing. And to be able to think about questions that you know are too difficult and to let them challenge you anyway is enormously important. I mean, it forces you to think well and to work hard, and it's okay if you're not the best person in the room at it. I don't think I was ever the best person in the room at math from the time I was 6 years old, because I always had my little brother around. So, but I enjoyed it, and it really set me up to move to genetics seamlessly later on. It did not set me up to have good hands. I had to be taught that by Alan Wilson and the people in the Wilson Lab. But it did set me up to think about problems logically. So one thing I would say to girls and boys is stick with math. Stick with science. It's okay to not be the best person in the room at it, but stay with it and keep working at it until you master one point and then master the next point. And the more you master, the more you will enjoy. At least that was true for me. Mm-hmm. And I think the other thing is that when you find a niche that you like, and for me that was genetics, and it was when I was a graduate student, you're all set. If you enjoy, right? If you enjoy the day-to-day work of doing genetics, then the fact that it takes a long time to have a result, that many experiments don't work, and that's perfectly all right for experiments not to work, that you're set. And we are incredibly privileged to be living in an era where there are great tools for doing science and where society values this. I mean, if only society valued poets as much as they value scientists. But we are fortunate. We are doing science. Yeah. So those are the two things. Do what you love and keep doing math and science until you feel like you've conquered the next step. Yeah.
I think that's good advice. I mean, traditionally, and I'll fall into that category, I also did math at high school, but I was not very good at it. Like you, I knew I wasn't thinking like a mathematician,
yeah, exactly.
But I did stick at it long enough to have some basis. Mm-hmm. But biologists in general have always said, oh, well, I'm doing biology because I can't do math. And of course, if there's anything that we've learnt, particularly currently, is how important mathematics, computational biology, physics, chemistry, all the sciences are to really understanding biology.
If you look at the people of our generation who created the Human Genome Project, a few of them come from medicine, but virtually all of them come out of math, physics, hard sciences.
Yes, absolutely.
It's a beautiful marriage of those things.
Yeah. And it's true today, and it's going to be true in the future. So those physicists who haven't gone to Wall Street have all ended up in biology. So
we make enough money. We don't need. What kind of measure is that? The only thing they should do is sponsor the Gairdner to bring all of us to Canada.
Right, there we go. We'll go and find some of those scientists.
We'll find them, right.
Just to give them some money.
Right.
So MaryClaire, thank you so very much for being with us today. I wish it were in person. We will meet in person, I guarantee. So really, it's been fantastic.
You as well, Janet. Thank you so much, and I look forward to seeing you in Canada some one of these days soon.
Absolutely. And so thank you, the audience, very much for being with us today. I'd also like to take a moment to thank our sponsors for their support for this event and for Gairdner Science Week as a whole. And please do check out the whole slate of Gairdner Science Week events. Visit the schedule section of Hopin or on our website at gairdner.org. And we hope to see you at more events soon.